CRISPR Technology by Zak Pham. But first of all, what is CRISPR? It's a new form of genetic editing, which stands for clusters of regularly interspaced, short palindromic repeats. Almost like scissors, for example, ones where you can specifically target and cut certain sites or parts of a genome to edit the gene. It proves to be a new form of genetic editing, rather than using transgenic methods, ones that involve foreign material used to implant certain traits into the subject, whereas CRISPR needs no introduction, specifically working within the genome to edit. The Cas9 enzyme is a crucial part of CRISPR, being those genetic scissors used along with a strand of RNA to target, bind, and cut the desired DNA site to allow for editing. CRISPR was discovered by Jennifer Doudna, a biochemist at UC Berkeley, and Emmanuelle Charpentier, the director at the Max Planck Institute for Infection Biology. Their discovery with CRISPR was renowned as rewriting the code of life. But why should we care? CRISPR is revolutionizing biomedical research, creating an easier and more detailed method to edit genes, helping us understand and treat genetic diseases, while also having some modern-day applications, being genetically engineered crops, animals, cleaner fuel, and overall help identifying and creating new treatments for cancers/diseases. All using CRISPR to make a better tasting, longer lasting, and more nutritional foods and crops resistant to herbicide and diseases, or even breeding and altering specific animals for certain physical attributes, for example, or to locate cancer-leaked genes to develop targeted treatments for mouth cancer. Some controversies that CRISPR has had so far has been mainly from He Jiankui, a Chinese researcher who was arrested and sentenced to 3 years in prison for creating the first genetically edited babies with CRISPR, producing twin girls and a third on the way, to apparently study and help prevent genetic disease such as HIV/AIDS. This instance severely violated the academic ethics and codes of conduct from his university, even involving the Chinese government, as well as charging him with illegal medical practice. This brought into question how rules and limits exist even in practicing and working with gene editing, where even Francis S. Collins, the director of the National Institute of Health, even stated on this case that without such limits, the world will face the serious risk of a deluge of similarly ill-considered and unethical projects. But yet, overall, CRISPR has continued to expedite and expand on biomedical research, revolutionizing our modern-day society, resources, and our own understanding on genomic editing today.